In this video, we'll discuss the structure of nucleosomes. So what do we know about nucleosomes? We know that nucleosomes contain a histone core that's made of 4 types of histones and a DNA double helix. The questions that need to be answered are, how do histone proteins interact with each other in the histone core? And how does the histone core interact with DNA? 2 heterodimers are formed in the nucleosome. On the left is a heterodimer between H2A and H2B. On the right is a heterodimer between H3 and H4. In both dimers, the interaction between the subunits is similar. Both interact via the histone fold domain in a type of interaction called a handshake interaction. The H3-H4 heterodimers assemble to form a tetramer within the nucleosome. This tetramer is stabilized by hydrogen bonds and hydrophobic interactions between the H3 subunits from each dimer. The H4 subunits do not contribute directly to the tetramerization. The H3-H4 tetramer assembles with 2 H2A-H2B heterodimers to form an octamer, which is the final histone core structure. This octamer is stabilized mostly by interactions between H2A and H4 and interactions between the 2 heterodimers, H2A-H2B and H2A'-H2B'. Let's look at the structure of the histone core from a different angle. This core is, in fact, a tripartite disc-shaped structure, where the middle is a tetramer that forms a central region which is sandwiched between 2 H2A-H2B dimers. The next question we need to answer is, what is the sequence of events in the formation of the nucleosome? The first step involves the H3-H4 tetramer interacting with the DNA, which then wraps around the tetramer. Next, the 2 H2A-H2B dimers attach on either side of the tetramer to form the nucleosome structure. In this configuration, the unstructured N and C terminal tails extend from the histone core, which facilitates internucleosome interactions and contributes to higher-order chromatin packing. The structure of the nucleosome was made clear in 1997. This marked a significant achievement in crystallography, as it was the first large complex to be resolved at high resolution. This work confirmed that DNA wraps around the histone core twice, forming 2 turns, and that the total length of DNA that's associated with the histone core is 147 base pairs. Next, let's discuss how histones and DNA interact within the nucleosome. The histone folds in each of the core histones are composed of 3 alpha helices, alpha 1, 2, and 3, and 2 loops, L1 and L2. In each heterodimer, whether it's H2A-H2B or H3-H4, the histone folds of the monomers are antiparallel. L1 on one monomer interacts with L2 on the other monomer, and the L1-L2 domains are on opposite sides of the heterodimer. The alpha helices, alpha 2, of each monomer cross each other. In a nucleosome, the histone-DNA interactions involve the sugar-phosphate backbone of the DNA and not the nitrogenous bases, and each heterodimer, H2A, H2B, or H3-H4, binds to 30 base pairs of DNA. There are 14 points of contact between the histones and DNA, about 1 in every 10 base pairs. The antiparallel orientation of the histone folds is critical for histone-DNA interactions in the nucleus. There are 2 types of DNA binding sites, alpha-1, alpha-1 sites, and L1, L2 sites. Let's first discuss the alpha-1, alpha-1 sites. We can see here that the alpha-2 helices point in the same direction, which is toward the DNA molecule. 3 major types of interactions happen with DNA at the alpha-1, alpha-1 sites. Alpha helices in biochemistry exhibit electrostatic interactions due to their dipole nature, where the N-terminal carries a positive charge while the C-terminal carries a negative charge. This facilitates electrostatic interactions between the positively charged N-terminals of alpha helices and the phosphate groups on DNA. This phenomenon applies to H3-H4 and H2B as well. There are also nonpolar interactions between the side chains of some of the residues on the alpha-1 helices and the deoxyribose group on the DNA. For example, the first turn of the alpha-1 helix in H3 and H4 forms a nonpolar interaction with the DNA. Finally, the third type of interaction is between residues with the side chains that contain basic negatively charged or hydroxyl groups. These form salt bridges and hydrogen bonds with phosphate groups in the DNA major groove. This applies to the alpha-1 helix of histones H2, H4, and H2b. The L1, L2 sites include the two loops at the N-terminal of the alpha-2 helix. In each dimer, you have two L1, L2 sites. And once again, there are three major types of interaction between the L1, L2 sites and the DNA. There are electrostatic interactions between the N-terminal of the alpha-2 dipole for all of the histones and the phosphate grooves from the DNA band. There are also hydrogen bonds between the DNA and nitrogen atoms of the histone polypeptide backbone. If you look at the structure of a protein, the backbone of the protein has nitrogen groups that are part of the peptide bonds. These nitrogen groups can form hydrogen bonds with DNA molecules. Hydrogen bonds occur between arginine residues in L1 of histones H3, H4, and H2A, as well as the hydroxyl group of threonine residues in the L2 loop of H3, H4, and H2b. These hydroxyl groups allow for the insertion of the arginine side chains in the DNA minor groove. These bonds define the orientation of the arginine residues in L1, which can be inserted into the DNA minor groove to form electrostatic interactions. Overall, you have these 14 points of contact between the histones and the DNA. The DNA double helix in a nucleosome is characterized by 5 different features. The first feature is that the DNA forms a superhelix. Supercoiling happens due to the wrapping of the DNA around the histone cores. And these superhelixes form 2 turns around each histone core. The second feature is that the interaction with heterodimer H2A, H2b, and H3, H4 distorts the path of the superhelix. The third feature is that along the superhelix, the twist number varies from 9.4 to 10.9 as a result of the distortion of the path of the superhelix. On average, the twist number is 10.2 base pairs per turn compared to an average of 10.6 base pairs per turn for free DNA. That means that the DNA double helix in the nucleosomes is overwound. The fourth feature of such interactions is that the overwinding of the DNA double helix impacts the relative position of the 2 turns of the superhelix. The 2 turns are said to be in register, which means that the minor grooves on each turn are aligned and form channels. Through these channels, the unstructured N-terminal tails can exit for the histone H3 and H2b. In this image, it is clearly shown that the N-terminal of histone H3 exits through the gap created by the 2 minor grooves of the DNA facing each other. The unstructured flexible ends of the other 2 histones, H2A and H4, interact with the DNA minor groove and contribute to the stability of the nucleosome. So we know that the double helix is distorted and that the twist number changes. This means that nucleosome stability could be affected by specific DNA sequences. And that brings us to the 5th feature, which is the presence of GC and AT-rich stretches in the DNA. Sequences that are rich in GC base pairs promote the bending of DNA, but only when these GC stretches are present in a major groove. Consequently, regions of DNA that are associated with the histone core typically have GC-rich stretches in the major groove, which faces the histone core. The AT-rich stretches promote DNA binding and induce a local increase in the twist number. These stretches are preferentially located in minor grooves that face the histone core and will promote strong salt bridges between histone residues and the phosphate group in the minor groove. All of these features that we've just described contribute to the stabilization of the nucleosome. In conclusion, histone cores form very compact structures that are stabilized by non-covalent interaction between histone folds, but also stabilize the coiling of 2 turns of DNA double helix through non-covalent interactions. The relative positioning of the 2 turns of DNA creates channels that are used by histone tails to exit and allow for the interaction of the histone tails between nucleosomes.